first part of this talk, I will give you an overview of bioimage analysis. Computers are great for many things, but object recognition isn't one of them, which is why contextual puzzles like this are used to confirm that you're a human online. While it is obvious to us which squares have traffic lights, a computer needs help translating binary digits or bits, which can be 1 or 0, into objects. And this is important to understand in terms of bioimage analysis, as you will see later. The goal in bioimage analysis is obtaining quantifiable information from microscopy images. For example, in these two images, you can visually tell that there is a difference between the control and the treated cells. But how can we describe this difference computationally and measure it? An example of how we might measure roundness is by working out the ratio of length by width. Using this metric, a perfectly round cell will have a value of 1 and a square will have a value of 0. We can then plot the roundness of the cells and demonstrate that treated cells are rounder than the control. In this talk, I will cover the general workflow involved in bioimage analysis, including what is required to generate suitable data for your image analysis. In many cases, researchers want to analyze images after acquiring them, only to find out that the images that they have acquired are not suitable for quantification. So I will cover the steps needed to generate images with good enough quality to properly carry out the analysis and end up with good results. Quantification should actually be the last part in the bioimage analysis workflow. The first step is to plan quantification and determine what needs to be measured. Then decide on a suitable imaging technique to carry this out, and your sample preparation will then be determined by the measurements you want to make and the microscope you are using. This planning should be done before the experimental aspect, which includes the acquisition and the eventual quantification. Now let me give you an example of this workflow. Let's say you want to measure the colocalization of two proteins in the mitochondria. Because the target structure is small and we want to measure colocalization, super resolution is probably the best option. This informs our sample preparation, and because we're doing super resolution, we're going to need a monolayer of cells. We will also need a negative control with no colocalization and a positive control with a high degree of colocalization. When the planning is complete, we can then carry out the experiment and begin acquiring our images and doing the analysis. So now let's consider a different scenario where we did not use the correct instrument and we did not have sufficient resolution to image the objects of interest. Then we would get images that do not resolve the objects of interest, in this case the mitochondrial proteins, and we would get the wrong result for our colocalization analysis. So sometimes we might not know what feature we want to measure because the actual biology may be unknown or we are unsure how to measure it. So we should always carry out a pilot study before undertaking a major experiment. Also, if you're unsure if a microscope's specifications will be suitable for your image, it's another good time to run a pilot study. To organize a pilot study, you can email us and begin the initial consultation.
An often neglected aspect of the bioimage analysis workflow is how to report and document the analysis you've carried out. As you process your images, you should be recording what you're doing in a lab book or saving templates and settings depending on the program. This is important for two main reasons. The first is reproducibility and the ability to consistently perform the analysis. And the next is publishing or writing up this part of your research, where you will need to provide detailed information. I will discuss this topic a little bit more later in the talk. A final point I'd like to make is to try and start your image analysis as soon as you can after acquiring your data. This is the best way of finding out if your images are suitable for the analysis you want to do and ensure you're getting the desired results. If not, you can then adjust the acquisition settings to something more suitable. Never plan to take all of your images and then expect to do the analysis. After weeks or even months of imaging, you do not want to find out at the end that they are not sufficient for the analysis you want to carry out. The only solution then may be to image them again or scrap that part of your research.
Now I will go on to the main part of the talk, introducing the general workflow in image analysis and how we go from images to graphs. So let's say we have a raw image like this. Generally, the workflow will involve pre-processing to correct for defects and improve the image quality, object segmentation, where we will identify the features we want to measure or define regions of interest, and finally, we will carry out measurements and do the data analysis. I will go through each of these steps in more detail in the following slides. Depending on the type of analysis we want to do, sometimes we can use a single program, and other times we'll use different programs for each step because we need some specific function. There are many programs available for image analysis. Unfortunately, this search engine and database were developed by the Network of European BioImage Analysts, which curates information about a wide range of programs. If you look at this window, you'll see that there are over 1,300 programs available as of January 2021. And you can also find reviews and papers on this site that will go into more detail about each of these programs. This table shows a list of freeware, free software that's available. However, if you're a new user, we recommend that you use ImageJ, Fiji, or Cell Profiler. In the BOMP platform, we provide support for a number of free software packages such as ImageJ Fiji, Cell Profiler, and Elastic. However, we also have licensed software packages dedicated to bioimage analysis such as Imaris, Huygens by SVI, Velocity, Metamorph, and Amira. We also support commercial software that is mainly designed for acquisition, but also provides some image analysis functions such as Zeiss, Leica, Nikon, Olympus, and the Harmony from PerkinElmer.